The overall goal of this procedure is to examine how tissue-resident cells influence the ability of vascular endothelial cells to support the recruitment of leukocytes during inflammation. This is accomplished by first establishing co-cultures of endothelial cells with mesenchymal stem cells, using one of two in vitro vascular models, the microslide model and the filter model. These cultures are then treated with cytokine prior to the flow assay. The flow-based adhesion system is then assembled in a step-by-step manner, with modifications made depending on the model system being used. The final step is to perfuse leukocytes over the endothelial cells and observe their recruitment from flow as well as their subsequent behavior. Ultimately, these multicellular flow-based adhesion assays have shown that crosstalk between mesenchymal stem cells and endothelial cells suppresses neutrophil recruitment during inflammation.
This method can help answer key questions in the leukocyte trafficking field, such as how do tissue-resident cells, for example mesenchymal stem cells, modify this process? Does this change in diseases such as chronic inflammation or cancer? Can we use this knowledge to help develop new therapeutic strategies?
To begin, establish co-cultures of endothelial cells with mesenchymal stem cells using the microslide model or the filter model as described in the text protocol. To assemble the flow system, first turn on the heater and set to 37 degrees Celsius. Then, attach a 20 ml syringe with the plunger removed, as well as a 5 ml syringe to a 3-way tap. Next, measure and cut a long piece of 2.4 mm tubing.